And welcome everybody to the February edition of the JSPS Pathological Grand Rounds, sponsored by the Shanks Foundation and the Pathological Society. As usual, the lecture will go on for around 45 minutes and there will be a question and answer session at the end. If you have questions, please put them into the Q&A box at the bottom of your screen and I will read them out at the end. Today's lecture is given by Professor Simon Leedham, and it gives me great pleasure to introduce Simon. Simon is a Professor of Molecular and Population Genetics and an Honorary Consultant Gastroenterologist at the University of Oxford. His research is into the morphogenic signalling pathways that control the intestinal stem cell in homeostasis, regeneration and cancer, and he has published more than 100 peer-reviewed papers in a range of top-notch high-impact journals, including Nature and other Nature-stable journals. Simon's research has been recognised by the United European Gastroenterology Rising Star Award in 2010, the British Society of Gastroenterology Francis Avery Jones Research Prize in 2015, and the Cancer Research UK Future Leaders Prize in 2017. Simon will give his lecture today entitled Stem Cell Adaptive Phenotype in Colorectal Cancer. Thank you very much indeed, Simon. Over to you.
Thank you so much, Mark, and thanks for the invite to join this really fantastic lecture series that you have here at the Pathological Society. I'm going to share my screen and hope that this works, and I hope you can now see the title and this rather long-winded title, Stem Cell Adaptive Molecular Phenotype in Colorectal Cancer, which we largely use so we could shoehorn it into the acronym STAMP on CRC. So I hope that my slides will now move. Yeah, great. I did my PhD in the rather wonderful pathological lab of Sir Nick Wright and back in 2004 to 2008. And during that time, we really subscribed to the somatic mutation theory of carcinogenesis, which really belongs to the idea that you have a kind of normal stem cell, which is exposed to a carcinogen of sorts and undergoes some genetic change, here represented by this yellow cross. And then that yellow cross bestows a fitness advantage upon that cell, which then is able to expand and grow quicker than neighboring cells, acquiring further mutations, such as the loss of this red chromosome here, and then which further expands further and faster, leading to transformation and progression. So really, it stems back from the original idea that there was a single cell of origin of any cancer, and that this sort of sequential accumulation of mutations was the process that led to initiation and transformation. And really, I think that sort of largely holds true, although subsequent work has obviously made, as with anything to do with cancer, has obviously made that a lot more complex. And really what we understand now is that there are these multiple clones within a tumor that expand and contract. So clones are not static, they're not fixed, they're dynamic, and clones will expand, other clones will diminish. But what you end up with at the time of tumor is really looking at along this line here, which is a really chimeric mix of multiple clones, each represented by different colored cells here. And then going on to the idea that actually metastasis may be coming from any parts of this different tumor, so that actually metastasis may be a sort of polyclonal seeding. So as like anything cancer related, the simplistic notion we had before perhaps isn't quite as true as it was. And really, a lot of the work from the Swanton stable has sort of proven this, or has at least looked at this. This is a really well-known paper from back in 2012 now, New England Journal of Medicine. This is a horrible renal carcinoma here. And Charlie Swanton and his team dissected out individual regions from around this tumor, and then undertook what was then fairly nascent whole genome sequencing technology to determine the genetic makeup of the genetic phylogenetic trees of each of these different regions. And fascinating, they were able to show that each of these different regions actually contain quite a different mix of genetic clones. So you had a truncal mutation, but then actually clonal evolution led to each of these regions having quite distinct genetic makeup, so quite profound regional genetic heterogeneity. And I think Charlie drew this tree because it looked exactly like this tree from Darwin's notebook, of which I think he is a sort of fully paid up fan member. The idea here is that you've got this sort of clonal evolution that's occurring within the tumor, and that's leading to genetic heterogeneity that spreads, that is across the tumor, and that genetic heterogeneity spells trouble. Why does it spell trouble? Well, the problem with heterogeneity is really adaption and resistance. And this is a really visual image of this from a paper in 2011 showing a BRAF mutant melanoma in a patient. Here is a BRAF mutant melanoma, end stage, as you can see, very cachectic looking patient. The lumps and bumps all over the skin here are metastatic melanoma. And this patient with his BRAF mutant melanoma tried a very novel drug at the time, a BRAF inhibitor, a new BRAF inhibitor, and had quite remarkable results. So this is the same patient on the back of being treated with a BRAF inhibitor. You can see that he's regained muscle mass, the lumps have almost all disappeared, his skin tone looks better, and actually everything looks remarkably improved. But very sadly, this same patient then developed resistance to the BRAF inhibitor. And this is the same patient in the third picture here, with recurrence of the melanoma, again, the cachectic appearance. And unfortunately, this patient died a short time after this photo was taken. And the really striking fact here is that mean time for drug development is about 9 years. It costs about 2 billion, and the BRAF inhibitors were probably no exception to that general rule. But the time taken for this tumor to develop resistance to that therapy was less than half a year, or about half a year. So we have this really rather sad fact that targeted therapies take a huge amount of time to develop and to get into patients. And actually the patient's cancers can evolve and adapt to that resistance within a matter of weeks. And when a sort of doyen of cancer biology, such as Bert Vogelstein, said this, resistance is therefore a fait accompli, the time to recurrence is simply the interval required for a subclone to repopulate the lesion. It really rather sounded the death knell for targeted therapies in their form at the time. So, but what we've been particularly interested in recently is thinking not just about genetic heterogeneity, because genotype is not the substrate for natural selection, it's phenotype. So phenotype is what Darwin studied when he looked at his finches in the Galapagos. As you can see, lots of different finches with lots of different beak types. Different beak types were either good for cracking nuts or seeds or for winkling insects out of wood. And really this phenotype of the beak in Darwin's finches is what he used as his model for natural selection. So phenotype is not just a function of genotype, it's a function of both the genotype and the environment within which the cell sits. And really we need to remember that, is that genotype is just one half of a big story. So Charlie's original paper looking at genetic heterogeneity at the time, really important, but perhaps not the whole story. So we've been asking the question recently is, can we find a molecular phenotype with which to measure colorectal cancer evolution? And to do that, we've been focusing in on our area of interest, which is the intestinal stem cell. And I'm afraid I'm going to have to tell a little bit about the intestinal stem cells, so bear with me here. So as we all know, the basic functional unit of the gut is the intestinal crypt. At the base of the crypt, we have this, in homeostasis, we have this very neat restricted stem cell zone right at the base of the crypt. And that includes some cells that are maintained here called the crypt-based columnar cells, here highlighted in red. So these crypt-based columnar stem cells give rise to daughter cells, and those daughter cells travel up the intestinal crypt through the transamplifying zone into the differentiating zone, and after 7 days into the poo. So these cells are pretty busy. They're turning over the whole time. And some beautiful work from the Clavers laboratory back in 2007, sort of revolutionary work when I was just finishing up my PhD, really spelled out how these cells are defined. And they're defined by being marked by a gene called LGR5, or Nick Wright used to call LAGR5. So LGR5 is the marker gene for these crypt-based columnar stem cells. And really, these are bona fide homeostatic stem cells, unquestionably. But actually, things have got a little bit more complex since then, because what we understand in regeneration is that the story isn't the same. When the tissue is damaged, the LGR5 stem cells actually disappear. The crypt-based columnar stem cells disappear for about 5 days. So therefore, the repair of the epithelium has to be done by alternative cells. And actually, there's been a huge amount of work sort of outlined here with some of these rather nice papers that show that there are a wide array of cells that are capable of returning to stem cell behavior and allowing the crypt to regenerate. So there could be Paneth cells, they could be secretory cells, they could be enterocytes, and even rare tuft cells are all capable when the regenerative milieu is changed, when the signaling within the regenerative crypt is changed, then these apparently post-mitotic cells can change their behavior and fall back to behaving like stem cells. So we have then a real range of cells, which we've called regenerative stem cells, a whole range of cells that are capable of behaving like stem cells when the signaling milieu is altered. So what we've done recently is ask, well, given that cancer is considered to be the wound that never heals, is there any evidence of these regenerative stem cells within cancer? And what we did to look at that was to take some signatures, some gene signatures for the LGR5 crypt-based columnar stem cells here, which are LGR5 positive, and Nexin-1, a marker here, negative, and SCAR1 negative. We took the signature of Munoz et al. From 2012 and used that to define these crypt-based columnar stem cells. Here on green cells are going to be representative of crypt-based columnar cells. But then we also took a signature from Yui et al., Kim Jensen's lab, to look for these regenerative stem cells. Here they're going to be highlighted in red from now on. These cells are LGR5 negative, Nexin-1 positive, and SCAR1 positive. And we took these two signatures and did gene set variation analysis to essentially lay these phenotypes across a sort of spectrum. So we've got a red regenerative stem cell enriched side on the right and a crypt-based columnar enriched side on the left, represented by green and represented by red. And we then defined what we call the stem cell index to try and look at the admixture of these different gene signatures within tumors to use as a surrogate of the admixture of stem cells within a tumor population. So we have here, within any tumor, you might have a batch of crypt-based columnar cells, you might have a batch of regenerative stem cells. Sadly, these things can move through stem cell plasticity, they can change their phenotype, and I'll talk more about that later. But really, we wanted to use the stem cell index to give us a sense of how many cells we had within an individual tumor. And this really is a measure of the admixture of stem cells. So I'll be using the stem cell index a lot throughout the remainder of the talk, but essentially it's this monitor of stem cell admixture within tumors. So the first thing we did was to look at some single cell data. This wasn't our data. This is from collaborator Sabine Tejpar. This is the stem cell admixture in normal crypts. So here we can see that there are lots and lots of green cells. These are the LGR5 crypt-based columnar cells, and very, very few cells with any regenerative marks at all. So in the normal homeostatic population, the crypt-based columnar cell is the stem cell. And I think that's fair to say that that's completely true. But in colorectal cancer, we've got a different situation. So here is a mixture of different colorectal cancers. Here again, the LGR5 crypt-based columnar cells represented by green, and you can see that there is a definite upregulation of those cells, increased number of those stem cells within a cancer. But there's also a really big population of these red cells, and these red cells are the regenerative stem cells marked by Nexin-1 and SCAR1. And you can see that there's sort of an equal number of regenerative stem cells as there are crypt-based columnar cells in the single cell data from these multiple patients, and relatively few yellow cells here, which are cells expressing markers of both crypt-based columnar and regenerative stem cells. So here we have two discrete and distinct populations of stem cells that are present in colorectal cancer, really sort of confirming the idea that in the disrupted signaling milieu of the colorectal cancer, we have the ability to form these regenerative stem cells. We have stem cell behavior with cells falling back to behave like stem cells where they shouldn't. So the next thing we did was to ask, can we see the stem cell molecular phenotype is altered in different human types of colorectal cancer? And I'm sure many of you will be familiar with the way that we molecularly subtype colorectal cancer. We use either the consensus molecular phenotype or this one, the intrinsic molecular phenotype, to look at different molecular subtypes of colorectal cancer. And I won't go on about these subtypes, except to say that the CMS2 subtype is the subtype most characteristically considered to be the adenoma-carcinoma sequence driven by APC mutation and further KRAS P53, whereas the nasty subtype is this CMS4 subtype This is driven by stromal activation, TGF-beta activation, various other things. And everyone will know, I'm sure, about the CMS1 subtype, which is predominantly the microsatellite unstable subtype. And as we can see, when we look across these different subtypes, there is a skewing of subtypes towards different ends of this spectrum. So you can see along here, we'll see a number of these graphs. This is the stem cell index along the x-axis here. Towards the left is CRIT-based columnar-enriched tumors. Towards the right is regenerative stem cell-enriched tumors. Here's the spectrum sort of visually laid out. And these CMS2 ones driven by Wnt signaling are skewed predominantly towards the left-hand side, the CRIT-based columnar LGR5 enriched side, whereas these microsatellite unstable and these stromally enriched tumors are more skewed towards the regenerative stem cell side. And that holds true for the intrinsic signature, where CRIS-B is the kind of equivalent of CMS4, and CRIS-A is kind of the equivalent of CMS1, with CRIS-C, D, and E being representations of CMS2 and 3. So again, skewing of the microsatellite unstable and stromally rich tumors towards the regenerative side, skewing of the APC-driven tumors towards the CRIT-based columnar side. And really, we really wanted, coming from a pathology or a very great interest in pathology, we really wanted to have a visual way of looking at this. So to do this, we used dual-ish staining. This is in situ hybridization for LGR5 in green and annexin-1, which is one of our regenerative stem cell markers, in red. And here we have this green-red dichotomy again, looking at tubular-villus adenomas, which are the classic kind of adenoma-carcinoma sequence. We can see that the tubular-villus adenoma is predominantly green. That really goes along with the CMS2 colorectal cancers over here. But serrated adenomas, traditional serrated adenomas, sessile serrated adenomas actually have quite a population of regenerative stem cells, as does IBD dysplasia, colitis-associated dysplasia. And as we can see into the cancers, the CMS1, 3, and 4 actually have quite a lot of red cells here, suggesting that they have a distinct population of regenerative stem cells. So we really wanted this to be a visual representation of the molecular index that our stem cell index was measuring. Now, I'm sure many of you know there are a lot of improving mouse models of colorectal cancer, really led by Owen Sampson, but with a consortium that we're part of the Cancer Research UK Accelerate Consortium, we've been able to share a lot of these mouse models around our community. These models are getting increasingly good. Here is a mouse primary tumor. I don't really need to say to pathologists that really looks like quite a decent representation of a human cancer. We also have these metastases that form, again, pretty nice representation of a human metastasis in the liver, the lymph node, and the lung. And really, we can divide different genotypes of tumor. These letters all stand for different genotypes. So A stands for APC, K stands for the KRAS, P53, and Notch, and various other abbreviations, which I'll come to. But as we can see, that these different genotype tumors have different molecular phenotypes that really do represent the human molecular phenotypes in this heat map. So we've now got a panel or a suite of mouse models that really nicely model the human tumor situation. So what we did was then take our stem cell index, we converted it to a mouse stem cell index, so similar genes, but just converted for mouse, and then looked across the mouse phenotypes. Again, I'll go through some of these genotypes. We've got the APC-min model. We've got an APC completely floxed. We've got APC-KRAS, APC-KRAS, P53 and TGF-beta, BRAF, P53 and Notch, KP is KRAS and P53, and KPN is KRAS, P53 and Notch. So we have these different mouse models, and applying the stem cell index, again, skewing towards the regenerative side or towards the CRIT-based columnar side, we can see that the phenotype of the stem cell molecular phenotype varies according to the genotype of the tumor. And interestingly, if you take organoids from these models and look at just the epithelium similarly is skewed in different directions. So this isn't just a function of taking immune or stromal cells out. This is an epithelial cell intrinsic change that ranges with the genotype of the epithelium. Again, visually, I like seeing pictures, as I'm sure many of you do. So we looked at these mouse models using our dual ISH stain. Here you can see an APC-min tumor, which is largely LGR5 green crypt-based columnar cells, moving towards an AKPT model, APC, KRAS, P53, TGF-beta, and a little bit of a range, lots of yellow cells there, lots of green, lots of red, moving towards KPN and BRAF models, where actually they are almost predominantly red regenerative stem cells. So again, we've got this phenotypic spectrum that we can measure both with molecular phenotype, but also with a morphological phenotype to show that there is this balance of stem cells. There is this dynamic equilibrium of stem cells occurring in tumors, and that equilibrium shifts according to the genotype of the tumor. So the next question we really needed to answer, and we had to do this for the publication, was, are both of these crypt-based columnar and regenerative enriched populations clonogenic? And that's a really important question in stem cell work. So to do that, we had to take the KPN mouse. We had to, KPN is KRAS P53 and Notch. We took that mouse because it's largely enriched for regenerative stem cells, although there are some LGR5 crypt-based columnar stem cells in there too. We disaggregated the tumors, we FACS sorted them for either Ephrin B2 as a marker of a surrogate marker of LGR5. LGR5 has no FACS antibody, unfortunately, so we used Ephrin B2 as a surrogate marker, and we used SCAR1 or Ly6G as a marker of the regenerative stem cell population. And we were able to FACS sort to enrich for either crypt-based columnar down here or regenerative stem cells up here. And we then plated those cells, single cells out, so single cells into organoid matrix. As you can see, when we FACS sorted, we had a large population of cells that were unmarked and a smaller population of both regenerative and crypt-based columnar type stem cells in the green, in the red and the green, respectively, with a much smaller population of cells expressing both. And when we plated those cells out from single cell, we could see that the regenerative stem cells here in red and the crypt-based columnar stem cells here in green, and indeed the cells expressing both markers, were all capable of forming clonogenic organoids, whereas cells expressing none were very unlikely to form clonogenic organoids. So what we have here is a population of cells expressing stem cell markers and showing that those cells are then capable of single cell clonogenicity, which is a sort of function, a marker of stem cell behavior. So having defined these molecular phenotypes, it was really important to try and understand which signaling pathways influence that stem cell phenotype. And to do that, we lined the TCGA tumors up. I'm sure many of you will be familiar with this. It's several hundred colorectal cancers with full genomic information. And we lined those tumors up along this spectrum here. So here we have on the y-axis, we have regenerative stem cells, population enriched for regenerative stem cells. And on the x-axis, a population enriched for crypt-based columnar stem cells in green. And we lined the entire TCGA along this axis and then took the decile that was most enriched for our two populations of interest. So we took our decile enriched for regenerative stem cells and our decile of tumors enriched for crypt-based columnar stem cells, and then cross-compared the mutations that were present within those enriched deciles. And what we saw was really something quite different. So within the crypt-based columnar enriched decile, a vast majority, something like 3/4, had APC mutations. So we had one, or the odd number here with beta-catenin, but almost all of them had ligand-independent, i.e. APC or beta-cat mutations in Wnt pathway, with very few, with no mutations in the Wnt pathway at all. Whereas what we saw in the regenerative stem cell enriched population was a range of other mutations. APC was relatively infrequent, and instead we saw ligand-dependent mutations like RNF43, some fusion mutations as well. So there was a difference in the mutation spectrum that affected the crypt-based columnar and the regenerative stem cell enriched populations. And then when we looked again at some of the other pathways of interest, MAP kinase and PI3 kinase and TGF-beta superfamily, again, you could see that some differences were being picked out. Here in the MAP kinase pathway, KRAS was present in both, but BRAF was exclusively seen in the regenerative stem cell enriched pathways. And in the TGF-beta superfamily, there was a few mutations across the spectrum in the regenerative stem cell enriched, but actually much more likely to have A-team-Q loss occurring in the CRIT-based columnar enriched decile. So what we were seeing here is that we were cross-comparing the comparison of mutations in some key pathways, looking at the different types of mutations in our enriched deciles, and showing that there were differences in the frequency of mutation selection within those two different molecular phenotypes. What we then did was do something very similar at a transcriptome level. So this is taking the RNA sequencing from the human and from the mouse, doing pathway enrichment, which I'm sure will be familiar to many of you. This is taking RNA sequencing and pulling out the different pathways that seem to be differentially expressed. This is the NES score from gene set enrichment analysis here. And you can see that we saw quite enrichment of similar pathways across both human and mouse, with the regenerative stem cell enriched pathways here in red and the CRIT-based columnar enriched pathways here in green, which, as you can see, were Wnt and LGR5 and things that you might expect. In the regenerative stem cell enriched pathways, we saw interferon gamma, KRAS, YAP, and TGF-beta coming up as our pathways that were enriched within both the human and the mouse tumors that had a predominance of regenerative stem cells. So we've got differential pathway expression, Wnt and LGR5 coming up in the CRIT-based columnar stem cells, TGF-beta, YAP, interferon gamma, all sorts of other microenvironmental stuff coming up in tumors which were enriched for regenerative stem cells. And really, that really fascinated us. So it's this idea that actually you've got epithelial cell intrinsic pathways like Wnt defining your stem cell population in the CRIT-based columnar population, but in the regenerative stem cell population, as you might expect from perhaps from tumors that are being driven by a regenerative milieu, actually we've got a load of external cell extrinsic signaling that seems to define a stem cell phenotype. So what we did here was to take our different mouse models. This is the APC-min mouse model, the AKPT, the KPN, and the BT, which sort of represent mouse models along this phenotypic spectrum, BT being the most regenerative, APC-min being the most CRIT-based columnar, and then looked at their immune, stromal, and matrix using multiplex imaging here. This is, and you can see the markers along the side here. I won't go through them all, but we looked for immune cell populations, stromal cell populations, and matrix proteins, and then defining genotype-specific differences between these different populations. So depending on the genotype of the mouse, then the stromal, immune, and matrix populations of cells were actually different. So there are genotype-specific differences in immune and microenvironmental remodeling. And actually, what we were quite interested in, given the pathways that we were identified, is that matrix is mostly responsible for YAP, stromal cells are mostly responsible for TGF-beta, and the immune cells are mostly responsible for interferon gamma. So we posited the question as, was it possible that actually immune microenvironmental stromal remodeling within our regenerative stem cell milieu was driving the epithelial phenotype in those tumors? So this is a complex box plot, which I'll talk you through it. So what we did then was really to start growing organoids of different mouse tumors. So we've got wild type organoids, we've got AKPT mouse organoids, we've got KPN mouse organoids. So APC, KRAS, P53, TGF-beta, KRAS, P53, and Notch mouse organoids. And then we added interferon gamma or TGF-beta to the organoid environment there. And what we do when we do that, when we add interferon gamma, we measure the stem cell index. Again, on the left here is CRIT-based columnar enriched. I'm sorry, I haven't written this in on this slide. CRIT-based columnar enriched. On the right here, it's regenerative stem cell enriched. And what we have here, when we add interferon gamma in wild type cells, is a profound shift between the CRIT-based columnar enriched and the regenerative stem cell enriched. So we have a shift in molecular phenotype through the addition of both interferon gamma and TGF-beta in wild type organoids. So if you stick external signaling onto wild type epithelium, you shift the stem cell molecular phenotype from a CRIT-based columnar molecular phenotype across to a regenerative molecular phenotype. Interestingly, if you do the same with AKPT, you can see that the basal level of the molecular phenotype is different for these different genotype mouse models. but there is still the same responsiveness to interferon gamma in AKPT and KPN mice. So even though you've got a sort of genotype specific change in your stem cell index, you can still manipulate that phenotype by applying external signaling pathways. And the only place where we couldn't change that phenotype is in the AKPT mouse model with TGF-beta. And that's because, of course, the TGF-beta is mutated, is gone in this model. So there is no way of transducing the TGF-beta signal to the epithelium. So what we have here, I think, and really sort of an important point, is that although the genotype may influence the underlying stem cell molecular phenotype, you can still shift that molecular phenotype with external signaling. So it's not fixed, it's a dynamic system. And to test mechanotransduction, so YAP is the key pathway involved in mechanotransduction. This is the pulling on the cells from the collagen and the matrix. So to look at that, we grew organoids in matrigel. We also grew them in collagen. In matrigel, we just have a population. When you do single cell analysis, you just have a population of CRIK-based columnar-enriched stem cells. But in collagen, you get this new population arising of regenerative enriched stem cells. So we have here a whole new population of stem cells that arise from simply growing organoids in collagen, where the collagen is pulling on the cells, activating YAP, which we can see here in this next slide. In the next bit here, we can see that YAP is activated in the collagen rather than not at all in the matrigel. So when you grow organoids in collagen, they'll pull on the cells, they'll activate the Hippo pathway, and that Hippo pathway then causes a population of regenerative stem cells to appear. So what we end up with is a sort of fitness landscape model. So we have these sort of two humps here. This is the phenotype space with a sort of a conceptual phenotype space, if you like. And we have up here on the y-axis, the fitness in this 3D model. And what we have really is two peaks that represent our CRIK-based columnar stem cells in green and our regenerative stem cells in red. And cells can move towards the CRIK-based columnar peak through acquiring an APC mutation. And cells will move towards the regenerative stem cell peak if they acquire YAP, TGF-beta, interferon gamma, or MAP kinase activation. And cells, unfortunately, I'm afraid, we think can move between these peaks through stem cell plasticity. So what we have here is a sort of really dynamic situation of cells being able to shift their phenotype according to the genotype that they have, but also the external signaling that they are surrounded by. And we use our stem cell index here as a marker of measuring that phenotypic shift. So the next question we really needed to ask was, is this stem cell phenotype in tumors fixed? And to do that, we took this really cool model here. This is an APC min tumor. So this is the mouse model of FAP. As you know, produces many adenomatous polyps throughout predominantly the small bowel. But what we attached it to was this moiety here called the LGR5 DTR. So this is wherever the LGR5 promoter is turned on, is attached to a diphtheria toxin receptor. Diphtheria toxin receptor can be activated with diphtheria toxin. And when that happens, cells that express the receptor die. So what this model does is kill all cells that are expressing LGR5 only when you give it diphtheria toxin. So we grew the APC, we generated the APC min LGR5 DTR model. In some of those animals, we then activated the diphtheria toxin. We killed all our LGR5 stem cells within the mouse. And then we looked to see what happened to the stem cell phenotype of the tumors over 24 hours and 5 days. And what we saw was really interesting. So when you take the APC min tumors, as we saw before, they're very strongly enriched for LGR5, very low levels of regenerative stem cells. But when you kill those stem cells using the diphtheria toxin, you can see we've lost almost all of our green stem cells here. And instead, what we've got is a very, very rapid upregulation of our red and Exin-1 stem cell population. So what is happening here is we've killed the predominant population of tumor stem cells, or of adenoma stem cells, I should say. And that has led to a profound shift in stem cell phenotype, with other cells, we think, returning to stem cell function, a whole batch of regenerative stem cells appearing. And interestingly, within 5 days, that population had then almost regenerated the green stem cell population. So what we're seeing here is this kind of shift in phenotype from down here on the bottom with this marker. We're moving from a crypt-based columnar population all the way across very rapidly to a regenerative population. And then over 5 days, that population then shifts back. So you've got this dynamic adaptive response to a selective pressure, kill your stem cells. Another stem cell population appears to allow the model, the tumor to continue. And that population then leads to recovery of the original stem cell population within a period of 5 days. So simply, tumors just adapt to the selective pressure that is put on them, roll with the punch, and recover their original population in the space of a week. And to look at that from really our fitness landscape model, what we did with our LGR5 cell depletion was squash, was kill the crypt-based columnar fitness peak, and cells simply adapted. They moved on to the regenerative stem cell fitness peak. So we saw a big increase in the regenerative stem cell population and a shift in the stem cell index. And then what happened is over 5 days, the regenerative stem cell populations changed their phenotype. They shifted their phenotype back to recover the crypt-based columnar stem cell population, and the regenerative stem cell phenotype shifted across to the crypt-based columnar phenotype in the space of 5 days. So this is this kind of incredible dynamic adaptive response of tumors to be able to roll with a selective pressure, an acute selective pressure, simply adapt and continue. And we really wanted to look for a human correlate of that. So we went to this fantastic study called the Foxtrot study. I'm sure many of you will be familiar with this. Foxtrot is a UK-funded trial to look at neoadjuvant therapy in human patients. And what happened is that they took a diagnosis biopsy and did RNA sequencing. They then gave 6 weeks of neoadjuvant therapy. This is in stage 2 and stage 3 colorectal cancers. So very unusual. They gave 6 weeks of neoadjuvant therapy and then took specimen 2 at resection off for RNA-seq. So what we have here is a series of specimens, specimen 1 and specimen 2, that surrounds the application of a therapeutic selective pressure. So a very unusual population in colorectal cancer. We do surgery early doors, and we never get that kind of temporal change. So we were really interested to see what happened in these tumors. And what we found is that there was a sort of dichotomy of tumors. So if we take specimen 1 here and specimen 2, the stem cell index, rather unhelpfully perhaps, is actually now on the y-axis. But what we've got is a population of tumors that don't particularly shift their phenotype. We called them static stem cell phenotype between diagnosis and resection. But we've also got a population of tumors where there was a profound shift in the stem cell phenotype between specimen 1 and specimen 2. So here we call this the plastic stem cell population. And those plastic stem cells moved in either direction, in both directions, but it was the shift in the stem cell phenotype that we were particularly interested in. So we've got some tumors that don't change on application of a selective pressure, and some tumors that do change their molecular phenotype on application of selective pressure. And I'll really just point you towards these pie charts at the bottom that showed that the static stem cell phenotypes, the tumors that hadn't shifted, had a much better better response to chemotherapy than the plastic stem cell phenotypes, where a large proportion of those had no response, histological response. So what we're seeing there is that if the tumor is capable of shifting its molecular phenotype in response to a selective pressure, then it's more likely to be resistant to the drugs that you give it. It's less likely to have an ongoing response to the chemotherapy. Therefore, the ability again of a tumor to adapt and respond to a selective pressure is associated with worse outcome for the patient. And I just want to briefly just touch on metastasis in the last few minutes, because it's a really interesting topic. What we're doing a lot of work on this from here on. What other people have shown, this is Jacco van Rienen's group, has shown that metastasis is largely done by the LGR5 negative regenerative stem cell population. So it's the red cells that metastasize, not the green cells. And really important. So you've got this tumor cell, you've got a primary colorectal cancer made up of red and green cells, but the cells that are found circulating around the body and then engrafting in the liver are the red cells. It's the regenerative stem cell phenotype that metastasizes. That population will then engraft within the liver, but in order for that metastasis to outgrow, it then has to regain its stem cell equilibrium. So it then has to regenerate the green cell population in order for a tumor to outgrow in a new host environment. So this is really fascinating here, this idea that it has to re-engineer its own microenvironment within a metastatic organ and allow it to return to a stem cell dynamic. And what we did was look at some mouse liver metastasis with these multiplex panels, and show that actually these very, very early tumors, these tiny tumors here in a mouse liver, you can barely see any metastasis there. In fact, the only reason you know it's there is because there's a huge pylon of CD4, green CD4 cells. So these CD4 cells are the only reason that you know that there are any metastasizing cells here in these micrometastases. But as time goes on, the tumors will start to enlarge, the secondary tumors will start to enlarge, they'll start to generate their own stroma, which is sort of similar to the stroma you see in the primary tumor and quite distinct from the liver stroma. And at that point, they start to extrude their CD4 cells. And in huge metastases, you can see the CD4 cells are largely completely extruded to the external side. And you've got quite a rich, complex periostin and alpha SMA enriched stroma within these very large metastases. And we can measure that using a sort of series of tools to show that the CD4 population declines over time as the size of the tumor increases, and the stromal compartment here in the blues increases. But what's equally fascinating is if you look at different tumors in the same mouse, so we have the occasional mouse here that will metastasize both to the liver and also to the lung. Here, this is a KPN tumor. This is the intestinal primary, very, very red in terms of its intestinal stem cell marks, interesting stroma and immune as well. But it's the stem cell marks I'll particularly point you to. The stem cell marks here are predominantly red. In the liver metastasis, there's a few green stem cells there. They start to recover their crypt-based columnar population, but actually in the diaphragm, the cells are predominantly red again. So what we've got here is a sort of different host organ requirement for the stem cell phenotype. So that depending on where you are, what organ you deposit in, the organ will help generate a stem cell phenotype that fits for where it is. In the liver, you need to have a kind of crypt-based columnar population recovered, but in the diaphragm, it's predominantly regenerative stem cells. And interestingly, we saw the same effect in human tumors. This is a shower of human metastases arising from the peritoneum. If those deposits fall on the sigmoid colon, those tumors are predominantly regenerative stem cell enriched. But if they fall in the small bowel on the serosal side, then they're actually predominantly crypt-based columnar enriched. So although these genotypes of these different metastases were all the same, this is all just from one patient, multiple metastases from one patient, actually, depending on the organ that those metastases land in, the stem cell phenotype in the metastasis is quite profoundly different. So what we have here is the environment of the metastasis changes the stem cell phenotype in a clonally identical population. So coming back to this fitness landscape model, in the primary colorectal cancer, you have a fitness landscape. If you move to the liver, you may have a slightly different fitness landscape. And if you move to the lung, then actually you're going to have a different fitness landscape again. So you have this sort of adaptive phenotype that shifts according to where it lands, according to the environment that it's in. So coming to the end, this really leads us to develop this intercompartmental coevolution model of colorectal carcinogenesis. And I'll try and explain what I mean by that rather complex title. What we have is within a normal population is two series of mutational pathways, either the serrated pathway or the conventional pathway. Each of the mutations that occurs within the epithelial cell of this model causes microenvironmental remodeling. So we're now trying to measure what each different epithelial mutation does to the microenvironment, but essentially the accumulation of epithelial mutation remodels the stromal immune and matrix microenvironment to suit the mutation that you've got. So it's sort of mutant epithelial cells build a microenvironment that suits them. That remodeling then leads to stem cell supportive cellular neighborhoods, which then feed back signaling in themselves, interferon gamma, TGF-beta, and YAP, to manipulate the stem cell phenotype, to manipulate the ideal stem cell phenotype for tumor growth. That then leads to metastasis, which then form their own stem cell supportive niche as well. So you have this kind of interdependence of the epithelium and the microenvironment, each one influencing each other, which I think is really interesting. What does that mean clinically? Well, what it does mean, we think, is that if you have a clinical phenotype and a molecular phenotype, unless we can measure the molecular phenotype, we're going to be missing a trick. So when we apply a selective pressure, such as chemotherapy, radiation, or in our case, LGR5 cell killing, we get this profound shift in the molecular phenotype without any real change in the clinical phenotype at all. So if we're just using scanning to measure tumor response to therapy, we're missing a whole lot of information that's going on at the molecular level. Sadly, there are then pathways that can become activated, such as TGF-beta, such as YAP, that are allowing the tumor to recover its molecular phenotype. So there is this sort of recovery phase. And what we really need to be doing, we think, is applying novel drugs to sort of inhibit these adaptive pathways. And then perhaps if we do that, we have the capacity to schedule our chemotherapy carefully and sensibly based on the measurement of molecular phenotype to enable effective tumor killing. So lots of information there. I'll try and summarize. Different populations of stem cells coexist in colorectal cancer. The stem cell phenotype is determined by the combination of both the epithelial mutations, but also really critically, the microenvironmental signaling pathways. The stem cell index can be used as a measure of this admixture, and we think that stem cell plasticity shifts this stem cell phenotype in response to selective pressures, and that really importantly, those adaptive shifts are associated with chemoresistance. So what we believe is that the stem cell molecular phenotype could be used to monitor neoadjuvant chemo response, allow us to develop a whole new load of drugs to inhibit the adaptive pathways, and really make us think carefully and titrate our drugs according to the molecular phenotype, which I think is far more responsive than actually the clinical phenotype that we measure at the minute, which is really based on cross-sectional imaging. So let me particularly thank the people that did this work. This was led by two fantastic PhD students in the lab, Nadia Nasred and Esther Gil-Vasquez, but was really an entire lab effort. And also to thank the funders, which include the Wellcome Trust as a fellowship I'm just finishing, and Cancer Research UK, who have funded a new program based on this. Lots of collaborators and friends across the country, and I thank you very much for your attention, and I will stop sharing there, I hope. Lovely. Thank you.
Lovely. Thank you very much indeed, Simon. That was an absolutely fantastic lecture, really well explained with some great data. Really good story, which I thoroughly enjoyed. Now, the lecture is now open for questions. I'd like to kick off a question, if I may. You showed the data, I think, from the Foxtrot trial, showing you had some patients that showed a pattern of plastic stem cells with dynamic changes, and others were static.
Yes.
Those changes. What makes the difference? Why are some patients plastic and why are some not?
That's a great question. So, I mean, what we think is going on, and it can only really be speculation. We haven't tested this. We are trying to do this in mouse models now, but we think that, I mean, Foxtrot is essentially standard chemotherapy. So it's epithelial, anti-proliferative, F5FU and oxaliplatin. So if some cells, i.e. tumors that are proliferating a lot, will respond more to that than tumors that aren't. So it's really about it's applying a fairly blunt selective pressure. It's all we've got at the minute, is the chemotherapy we have, but it's applying a relatively blunt selective pressure in an unselected way to a population of tumors. What would be really fascinating, and what we're testing in the mouse models now, is whether we can apply much more targeted therapies based on the molecular phenotype we have at diagnosis. So if you have a regenerative stem cell phenotype predominantly, perhaps you do better with a MAP kinase, a BRAF inhibitors and MAP kinase inhibition in the first instance, rather than the rather blunt tool of FOLFOX, which is really only going to target your proliferating epithelium. So I think, again, speculative, and we're trying to do the work to prove it, but I think that's to do more with the bluntness of our chemotherapeutic armory at present than it is necessarily. It's probably a differential response of the tumor to the bluntness of the tool.
Great, thank you. So can I encourage audience members to put their questions in the Q&A box, please? And we do have a question in the Q&A box, and it's on the same theme as the Foxtrot trial, if I can read it out. This is a question from Vinaya, who asked, "Thank you for such an excellent talk. In the Foxtrot trial, did you see a difference in response based on the pre-treatment enrichment for CBC versus RSC signatures? If there was a switch during treatment from CBC to RSC, or the other way around, Was there a difference in treatment response?
Yeah, it's another very similar question to yours, Mark. I mean, it's a really great question, Vinay. Thank you. So I think probably I partly answered that, but let me also just point out that in what we didn't see is any impact necessarily of the index stem cell phenotype on outcome. So if you just take the stem cell index at diagnosis as a one-off measurement, it's not particularly associated with tumor outcome, with life, with ongoing life and recurrence. So the stem cell index per se is not a very good predictor of outcome per se. Where we think it's useful is really in looking at that dynamic shift. So it's the dynamic nature of the stem cell index that I think is going to be important. It's not just another CMS or another CRIS that gives you some prognostic indication. It's really going to look at how tumors respond to therapy. And I think, based on Vinay's question, where we think we could use it is really to start titrating drugs. So we have a whole range of new drugs in colorectal cancer. TGF-beta inhibitors look really interesting. The BRAF combinations look really interesting. What I think we should be doing, and what I would hope that we can do in the future, is move towards a neoadjuvant setting where we can actually start to titrate our drugs against a measurement of molecular phenotype. If we can do that, if we know what's happening at diagnosis, if we know what's happening post-treatment, we can say, well, that drug worked really well in that population of cells, and that drug worked really well in a different population of cells. So I think it's really starting to use the measurement to titrate drug effect that we will hopefully see the biggest benefit. But that's a great question, Vinay. Thank you.
Thank you for that answer. I have another question for you, and that is, when you were looking at the gene mutations and also some of the expression signature patterns as well, you showed that mutant KRAS tended to be present in both of the signatures for the two stem cell types. But when you looked at BRAF mutation, that showed a strong association with the regenerative stem cell type. Yes. So what's the explanation for?
Yeah, it's a great question again. Who knows? Yeah, absolutely. I mean, so what we did see when we lined up tumors by increasing mutation, so by cumulative mutation, so both in the mouse and sometimes more difficult in the human. But if you put APC into a mouse, it predominantly causes a shift towards the CRIK-based columnar phenotype. If you then add KRAS, that causes a very obvious shift towards the right. So KRAS per se shifts towards the right, and KRAS driven initiated tumors are very, very regenerative. So I think KRAS or MAP kinase activation per se causes a shift to the right in the stem cell index. So it enhances regenerative stem cell phenotype. But BRAF was really only present in those tumors that were very enriched for regenerative stem cell phenotype in the first instance. I think personally that's got to do with the fact that most BRAF mutations will be present in the serrated pathway, and the serrated pathway, the cell of origin is not the LGR5 stem cell, it's this metaplastic population of cells. So there is already a kind of regenerative inflammatory drive in serrated pathways at initiation, and I think that essentially BRAF is selected in those because it enhances that regenerative nature, activates YAP very early. But again, why KRAS and BRAF should behave so differently is a question I can't answer. It's a great question.
Thank you. Now we have another couple of questions in the box. Let me ask you a question from Hema. Do the RSC interact more with their microenvironment compared to the CBC ones, since RSC seems to be the main one with metastatic potential?
Yeah, really, I mean, great questions from people here. Thank you so much. So that's really another fascinating idea. So it's certainly the case that the regenerative stem cells are driven by the microenvironment. So the three pathways we think are most important are interferon gamma, YAP, and TGF-beta. So those are all coming from the microenvironment, whether it's from the matrix, from the immune cells, whether it's from the stroma. So I think it's probably the regenerative stem cell is driven by the regenerative or the inflammatory milieu. Now, why those cells are more likely to metastasize is a great question. That's work that Jacco van Reenen did using intravital endoscopy, microscopy, to show that it's the regenerative stem cells that actually invade. Again, because of the plasticity of this system, it's really difficult to know whether or not those regenerative stem cells are actually more dangerous as such. It's possible that as a CRIK-based columnar cell in the middle of the tumor, if it migrated to the edge of the tumor where it's surrounded by its stroma, then perhaps it's going to start to behave in a different way. This is a dynamic system, I'm afraid. And it's those sort of tumor buds, that you guys spot in our pathology samples. Those are probably CRUK-based regenerative stem cell populations that are budding out, surrounded by stroma, and are behaving badly. Philip Dunn is doing some nice work now to look at those, at the phenotype of those tumor buds.
Thank you. Another excellent answer. Okay, so the next question is from Joseph, who asks, you have demonstrated several activation pathways that influence the shift from regenerative to CBC stem cells. Are these independent, or do we think we may block all of them for effect?
Yeah, again, really important. So we're doing the program that we've just been funded to do is really to try and pin down what are the key signaling hubs here. We're going to go for YAP and TGF and interferon as our three earliest targets. We don't know yet which is the kind of the core, the nexus of that capacity. YAP seems to be promising because it's really downstream of all sorts of other pathways as well. But there is, arguably, we may need to identify which are the adaptive signaling pathways, and then titrate our chemotherapy in those to be able to say which is the most effective. So there's a lot of work to do in this space, Joseph. And I think probably a systematic approach to doing it, because and that's where our mouse models come in. Yeah,
absolutely. Okay, so another question from Tao, who says, can computational modeling help understand the temporal spatial changes of stem cell genotypes, phenotypes during treatments?
Yeah, really. It's another really interesting question. So we've not done any particular sort of mathematical modeling of this. I suppose we are starting to do a bit of organoid work in this space, where we're using a very nice new sort of phenotypic measurement tool of organoid change in shape. So we can start to apply things like drugs that activate YAP in organoids, and we see a very profound difference in the organoid shape and behavior once we've done that, which we can then correlate with stem cell molecular phenotype using single cell sequencing. So I think that we're just starting to talk to the mathematicians in Oxford now about using that as substrate for computational modeling. But Tao, if you've got any ideas in that space, then please do drop me a line.
Good suggestion, thank you. So we have time for one more question before we finish. And this is on a subject close to both your heart and my heart, Simon. This is a question from Jonathan, who says, does this way of thinking shed any light on the specific etiology or pathogenesis of dysplasia/neoplasia in chronic inflammatory bowel disease? It seems that cells in that context would be moving in and out of the regenerative crypt stem cell phenotypes.
Yeah, brilliant question, right on the money. Nadia, who was my PhD student and Mark very kindly examined recently, has now moved to Glasgow, is sort of continuing this work. So we think that the regenerative stem cell phenotype, in comparison to sporadic bowel disease, inflammatory bowel disease dysplasia has a predominance of regenerative stem cell phenotype. That fits with this idea that APC mutation is particularly rare in those tumors. They do acquire Wnt mutation, but they do it through the ligand-dependent type of Wnt mutation that we see in the serrated pathway. So yes, I would argue, I think that the regenerative milieu of the chronically inflamed bowel enhances a population of regenerative stem cells, which are then, of course, subject to mutation. So the cell of origin, I think, in inflammatory bowel disease is not the LGR5 stem cell. It's more likely to be these regenerative stem cells.
Thank you. Another great answer. Now, we do have a tradition with these pathology grand rounds lectures that we finish on time at 10 o'clock to give the whole morning back to people. So can I end by thanking you once again, Simon, for an absolutely fantastic talk, really, really interesting material, and also for a lively and interesting question and answer exchange at the end as well. So
thank you to the great questions, and to you, Mark. That's really nice to have such interesting questions.
Thanks very much, and we'll sign off there. Thank you.
Thank you.
Bye.